This is the fourth and final part of the first lecture. We're going to talk about brain cells. The two primary types of brain cells are neurons and glial cells. So neurons are responsible for the information flow that is ultimately the primary function of the brain. Glial cells have many functions. They're primarily support-oriented, but please don't think that they're any less vital. You would not survive or have even developed if you did not have glial cells. They help stabilize and direct neurons, especially when the system is developing. So for example, the way when you were a fetus and your central nervous system was developing and your brain connected to the entirety of your body, the way in which it connected to the right parts of your body was controlled by glial cells. It also performs various maintenance functions for neurons, keeping them in place, keeping them fed, such. They create myelin sheaths, which we will talk about in just a second, which actually help speed up neuronal transmission. They clean up plaque and the waste that is generated by neurons, and new things are being discovered all the time, so there are probably more functions to glial cells that we do not know yet. The basic structure of a neuron, it can be classified into three parts. The first is the cell body. This is the sort of round structure at the top that you can see. It contains the nucleus, which has the genetic information of the cell, along with primary cell components that allow it to perform its basic living functions. Around the cell body, you can see what are called dendrites. These look like little tree branches or roots, and these are what receive connections from other neurons upstream. And the axon is this long tail portion of the neuron, which at the end has axon terminals, which are again branching, and these pass information down to the downstream neurons. These little blue nubs along the axon that you see, these are myelin sheaths. They're generated by glial cells, and they allow signals to travel at a much higher rate than if they did not exist. When you get a condition called MS, it is actually a degradation of the myelin sheaths, and this causes trouble in our ability to transmit information throughout the central nervous system. It is important to note that signals only travel in one direction. They are registered at the dendrites and travel down through the axon and are passed on via the axon terminals. They will never flow in the other direction via axon terminals to dendrites. That does not happen. An interesting component or quality of neurons is that the length of axons can be very variable. Some of the cerebral cells are very, very tiny because they don't have to transmit information very far, literally from one cell to an adjacent cell, and they can be as small as 60 microns. A micron is 1/1,000,000th of a meter. Other ones can be very long, like if you imagine the ones that go from the base of your spinal column all the way down to your pinky toe. Some of those can be as long as a meter. And in some other creatures, they can be longer still. They are neurons and their axons that travel the entire distance down a giraffe's neck and then from its back all the way to the bottom of its feet. And those are almost as long as we are, which is pretty wild. Note that they are single cells, right? It's one cell that is that long. Okay, information moving from neuron to neuron. So neurons are not directly connected to one another, and instead there is a tiny space between each of them. This space is called a synapse, and it's unbelievably small. Now, sometimes you'll see depictions of it in textbooks, and I will show you a picture on the next slide, and people tend to assume it's a little bit larger than it actually is. So for reference, if you were to stack 4,000 synapses approximately end to end, those would be about as thick as a standard sheet of paper that you get when you print out an assignment. Now, the neuron whose axon terminals are coming to the synapse, the one who's passing on information, these are called presynaptic neurons. And the neuron whose dendrites are at the synapse are called postsynaptic neurons, meaning the ones that are going to receive the signal or are downstream. The way in which information travels through the synapse is via chemical signals, and these are called neurotransmitters. They are oftentimes molecules of various different sizes. Speaking of neurotransmitters, over 100 different kinds have been identified so far, even though we don't know all of the functions of each of the ones that we have identified. Now, in this image, you can see a picture representation of a synapse and the neurotransmitters passing across it. You'll see that this is a zoomed in portion to the connection between one of the axon terminals and one of what are known as dendritic spines. So before anything happens, you have these little sacs, if you will, inside the end of the axon terminals, and these contain all manner of different types of neurotransmitters. Now, when a signal travels down the axon, which is called an action potential, just in case, the neuron will release some of these neurotransmitters. And the way that happens is one of these little vesicles that are containing the neurotransmitters will migrate down to the very bottom, and once they make contact with the cell membrane, they will actually open up a little hole and just dump out the neurotransmitters into the synaptic space. Now, these little neurotransmitters will just kind of move across the space. Their motion is ultimately random, but again, because a bunch of them are released and the space is so small, they will ultimately go and bind to specific receptor sites on the postsynaptic neuron. Now, there are different sites for each of the neurotransmitters, each being specific to their connection. A good way of thinking about it is keys and locks. Many keys all look similar, but each of them only work in a specific lock. And that's how neurotransmitters work as well. An interesting side note is many of the drugs that we take, they work on us because they are molecules or contain molecules in them that very closely resemble or are not, or could be identical even to certain ones, to our neurotransmitters. So for example, when we take some kind of morphine because of pain, or heroin for that matter, it is actually modeling one of the opioids. And opioids are actually naturally produced in the body when it takes significant trauma. It's just that when we take a dose of morphine or heroin, it's on a much larger scale. This is also how we use certain medications to treat people. So if you have ever taken a depressant medication or an antidepressant, sorry, oftentimes they'll use things called SSRIs, or selective serotonin reuptake inhibitors. And what that is doing is when serotonin is released, because that is also a neurotransmitter, into this space, what those will do is they will keep it from being reabsorbed. Because when neurotransmitters are released into this space, they don't just stay there forever. Most of them are either absorbed by the postsynaptic membrane, but many of them will then just be reabsorbed again by the presynaptic membrane. They will be encased once more and then be used again once a specific signal comes down the axon. They won't just stay there forever. Now, an easy way for you currently in this class to classify neurotransmitters, just to make things simple, I guess, is they can either be excitatory or inhibitory, and they can either have a short duration or a long duration. We will spend most of our time focusing on the former of excitatory versus inhibitory. What we mean by this is that an excitatory interaction or set of neurotransmitters will cause there to be some type of activity in the postsynaptic neuron. Inhibitory ones will prevent it from taking part or activating, which I will talk about a little bit more now on the next slide. When it comes to neuronal firing, this is when neurotransmitters bind to the receptor sites on dendrites of the postsynaptic neuron, and these change the electrical potential in the cell. This just means the current state of charge within that cell. And if this electrical potential passes a certain threshold, then that cell will fire. And whenever we're talking about a neuron firing, we just mean it's passing a signal down the axon. Now, neurotransmitters can be passed from many neurons into a single neuron, or they could go from one neuron that is connected to many other neurons, And a lot of the time it's actually somewhere in between, where you have entire clusters of neurons that are then connected to even more clusters of neurons. And as you saw with the little axon terminals and the dendrites, these toy pictures do not do justice to truly how many they are. You can think of this as the analogy of when a child draws a tree versus how many branches and leaves are actually on a tree. Now, what happens when excitatory and inhibitory neurons come into play is inhibitory neurotransmitters ultimately increase this threshold, meaning a neuron needs to receive a greater amount of signals from other neurons before it will fire. Excitatory neurotransmitters will decrease this threshold, making it such that it needs to receive less information from other neurons before it will itself fire. Now, it is also important to note that it is not one or the other. It is often the case that you will have a neuron that is receiving signals from multiple different neurons, and some of those neurons will be passing inhibitory messages and the others will be passing excitatory messages. And this is ultimately an additive process, where you could just think about it as you add all the excitation ones to all the inhibitory ones, and whichever one ultimately wins out will determine whether that cell fires or not. Another important thing to remember about neuronal firing is that the magnitude of the electrical signal, this is the amount of voltage that is generated and then the current that flows down, is always the same. No matter how much input it receives or how little, as long as it passes that threshold by a tiny bit or by a huge margin, it will always pass down the same amount, the same magnitude of current. And this is called the all or none law. The way in which neurons communicate complex signals is one through the way in which they fire, their frequency of firing. Do they fire many times a second or only a few times a second? Do they fire continuously or do they fire intermittently? These patterns of firing are one of the ways in which they will communicate different types of information. Further, the type of neurotransmitter that is then released into the synapse is another way in which different forms and messages are able to be passed, because each neurotransmitter ostensibly has a slightly different function. And it is this combination of multiple frequencies and multiple neurotransmitters that allows for such complex signals to be passed through the brain, even though an individual neuron is considered to be dumb because all it can do is fire or not. And I guess that leaves you with an interesting thing to ponder now of if you do have this vast collection of individual neurons that basically aren't intelligent unto themselves and all they really do is turn on and turn off at different rates, how from that do you get all that is you?